Hello bachcho. Let's see the question. So the question says second letter of the name restriction endonuclease came from first option is genus of organism, second is species of organism, third is family of organism and fourth is class of organism. So इन चार ऑप्शंस में से हमें करेक्ट ऑप्शन को आइडेंटिफाई करना है. So the key concept in this question is to first understand what is restriction endonuclease. So this is an easy question. Let's begin by first understanding the key concept. So restriction endonucleases, also known as restriction enzymes, are enzymes that can cleave DNA or cut DNA at specific sites only. So restriction endonuclease एक type of nuclease enzyme है. So nucleases are responsible for cutting or cleaving DNA. Now restriction endonucleases are specifically responsible for cleaving the DNA at specific sites only. अब restriction endonuclease can recognize a sequence of nucleotides. And ये जो sequence of nucleotides which is recognized by the restriction endonucleases are known as restriction sites. So restriction sites को restriction endonuclease recognize करते हैं और फिर वो cleave करते हैं उस DNA को into fragments at this particular site only. So every restriction endonuclease has its own restriction site which is recognized by it. Now, this restriction endonuclease was first discovered in bacteria. So this is a protective mechanism which is present in bacteria against bacteriophages. So the restriction endonucleases which are present in bacteria are particularly specific for the bacteriophage DNA, and therefore it can only act upon the bacteriophage DNA and cleaving them into DNA fragments so that it is no longer harmful for the bacteria. अब हर restriction endonuclease का एक particular naming pattern होता है. So there is a nomenclature for every restriction endonuclease. So अगर हम example ले of echo R1, then so now if we see how the naming of echo R1 takes place. So first of all, ये जो echo है वो stand करता है for the bacterial species from which this enzyme is derived. So basically echo R1 is derived from Escherichia coli. So the first letter comes from the genus name, that is Escherichia. And the first letter E is for the name for the restriction endonuclease. Then the second two letters comes from the species name from which this enzyme is derived. तो ये species कौन सा है? Coli. So Escherichia coli से since हम ये enzyme derive कर रहे हैं तो ये second two letters हो गया को. अब third है strain. So कौन से bacterial strain से हम ये enzyme derive कर रहे हैं उससे हमने first letter लेना है. So the first RD strain. So the RD strain of Escherichia coli is the strain from which we derive this enzyme. So इसीलिए इसका first letter यहाँ आ जाएगा and the last number जो assign किया गया है इस enzyme को is the number or the discovery. So which enzyme was discovered first? Now since echo R1, so echo R1 was discovered first from the Escherichia coli RD strain. Therefore, the enzyme is given the number 1. So from here we get the name echo R1. So the first letter E, Co, R and 1. So that is why the name of the enzyme is Echo R1. So therefore we can see that the second letter, so this is the second letter of the name of the restriction endonuclease, and we see that the second letter of the restriction endonuclease is given by the species name from which the enzyme is derived. So in case of eco R1, the second letter is C, which is derived from the coli, that is the species of the bacteria from which echo R1 is derived. So now if we see the options, then the first option says genus of the organism. So we saw that the first letter is derived from the genus of the organism. So this is incorrect. The second option says species of the organism. So we saw that the species of the organism was derived from the second letter. So this is the correct option. Other options, that is the third option, family of organism. So family of organism is not considered in naming of the restriction endonuclease. So again, this is incorrect. Also, the fourth option, that is the class of the organism, is also not considered in naming of the restriction endonuclease. So again, this is also incorrect. So the correct answer out of the four given options is option number 2. So our final answer for the question is option number 2, that is species of the organism. So the second letter of restriction endonuclease is derived from the species of the organism. Hope you understood this well. Best of luck. Thank you.